Hello everyone, my name is Angelina, and today we'll be learning about the brain-computer interface, debunking the conception, number 1, the brain can run on a computer, and number 2, computers can run on brains. So firstly, in order to consider whether computers can run on brains, we need to learn about brain organoids. Brain organoids are in vitro three-dimensional aggregates that are created through the self-organization and differentiation of human pluripotent stem cells, which are the base cells for all cell types. In order to make these, scientists use a cocktail of chemicals, and then these stem cells can become brain-like tissues that can recapitulate aspects of a developing brain structure and function. After the cells have differentiated, they can form neurons with varying complexity and density depending on the type of organoid and how long it has been cultured. At synapses, the neurons act the same as they would in the brain, releasing various neurotransmitters that bind to their respective receptors. Neurons and organoids can also generate action potentials, which are rapid changes in the electrical charge across the neuron's membrane. This occurs in response to external stimuli or spontaneously due to the neuron's inherent properties. Action potentials travel along the axons to the synapses, where they cause the release of neurotransmitters. These chemical messengers cross the synaptic gap and bind to receptors on the postsynaptic neuron, triggering further electrical changes and propagating the signal. Scientists use techniques like patch clamp recordings or electrodes to measure the electrical activity of neurons within organoids. They can also measure calcium concentrations with staining methods, visualizing changes in intracellular calcium levels associated with neuron firing. Over time, these cells mature and self-organize into structures that resemble different regions of the brain, such as the cortex, hippocampus, or even the retina. The organization includes the formation of distinct layers and regions that display some degree of functional specialization. These mini-brains provide a physiologically and psychologically relevant in vitro 3D brain model for the study of neurological development and disease processes that are unique to the human nervous system. They have important applications in studying neurodegenerative and neurological disorders such as autism, schizophrenia, or brain defects caused by Zika virus infection. So we can now glean into a little bit more about how organoids are being applied in the field of modern science and research through our Brain Aware trial. So a group of Johns Hopkins University scientists set out to model unsupervised learning through brain plasticity using brain organoids. The team constructed Brain Aware by mounting a functional brain organoid onto a high-density microelectrode array to track the electrical activity. The human brain organoid for Brain Aware was characterized by various brain cell identities, for example, mature neurons and astrocytes, and early development of brain-like structures, for example, ventricular zones. As electrical stimulation pulses were applied to Brain Aware, they evoked neuronal activity and demonstrated representative nonlinear response to pulse voltage, which is super cool because it kind of implies that the brain organoid acts in a very similar manner to a regular brain within a human. Another research team led by Feng Yu and his team at Indiana University Bloomington also generated a brain organoid from stem cells attached to a computer chip and connected their setup, a rendition of Brain Aware, to an AI tool. The brain system was able to execute mathematical calculations, play video games, and show rudimentary speech recognition. 240 audio clips of 8 people pronouncing Japanese vowels were presented to the organoid as electrical signals. Then the organoid responded and generated signals in the neuronal networks of its brain organoid, and this was fed back into the AI tool for decoding. The organoid recognized the clips with 78% accuracy, but it is unclear if this was stored in any memory. Through the information processing model for memory, we know that in order for memory to be encoded and stored, the brain must first experience a sensory signal, in this case, external electrical pulses. Then this must be stored in a sensory memory, which can then be stored in short-term memory, which eventually can be stored in long-term memory. However, memory storage in response to a single stimulus exposure, known as one-shot learning, is a fairly new field of study, which I believe would be directly pertinent to this experimental trial. In biological systems like the human brain and potentially in brain organoids, storing a memory from a single exposure involves several robust molecular and cellular processes, including rapid synaptic changes through long-term potentiation, a process where synaptic efficiency is increased. This is typically achieved through the rapid influx of calcium ion channels into the neuron, leading to the activation of kinases that enhance the strength of the synaptic connection, rapid synaptic changes through immediate early genes, which are genes that are quickly activated by neuronal activity, and rapid synaptic changes through neurotransmitter modulation, a potent singular stimulus, which can lead to the release of neurotransmitters and modulators that enhance synaptic plasticity. So, despite being able to integrate all of this cool technology with organoids, brain organoids are contentious for several reasons. Firstly, brain organoids use human bodily material. Secondly, tests with organoids are difficult to replicate. And thirdly, they are unfairly regulated and pose potential ethical and moral quandaries. Unsurprisingly, problems of unwarranted expectations and misconceptions arise with the notion of mini brains, or brains in a dish. Even the term organoid employs strong similarities regarding features, cell types, and functions of the respective organoids that are not present in most models. While a resemblance to the human brain commonly justifies their use as models in brain research and ethical discourse, these similarities have raised concerns that organoids might develop sentience or consciousness, which is definitely problematic. So now moving on, we're going to talk a little bit about the second conception, which is brains running on computers through Elon Musk's Neuralink. Neuralink is an implantable brain-computer chip that intends to create a generalized input and output platform capable of interfacing with every aspect of the human goal. The first indication they aim to address is the restoration of digital autonomy to people living with quadriplegia or amyotrophic lateral sclerosis, which is ALS. Quadriplegia, also known as tetraplegia, is a condition where there is paralysis below the neck. It is typically the result of a spinal cord injury, which may occur due to accidents, falls, sports injuries, etc. Sometimes these conditions can also occur due to brain damage in the motor cortex. Both result in the brain's inability to communicate with the extremities of the body, losing sensation, motor control, and autonomic functions. ALS is a progressive neurodegenerative disease that affects nerve cells in the brain and the spinal cord. Motor neurons deteriorate gradually, leading to muscle weakness, loss of motor functions, and eventually paralysis. The cause of this disease is unknown, and the loss of these neurons primarily affects the motor cortex and movement-related brain functions. Neuralink works by acquiring electrical signals from the brain via electrodes implanted across the brain. The device processes the wireless and wirelessly transmits the neuronal data to an instance of the Neuralink application running on an external device, such as a computer. The Neuralink application decodes and translates the neuronal data into actions, such as moving of a cursor on a computer screen. The first procedure was conducted earlier this year on a patient with quadriplegia. The patient was able to successfully control movements of a computer mouse, playing various video games and chess through his mind. However, some of the electrodes became loose in the patient's brain, almost leading the company to remove the device from the patient's brain. In my view, this experiment and device were morally and ethically wrong for several reasons, including that the patient's data and privacy of future users' privacy is significantly breached and effectively stored on an external surface, which can be very easily reviewed and manipulated by others. This is a very scary thought. Also, completely seamless communication between humans and computers is also worrying. We are already spending so much time on electronic devices disconnected from one another, and this device practically guarantees the extinguishment of human-to-human communication. There's also the concern regarding an individual's autonomy under this device. Do we really know whether or not the device is influencing an individual's emotions, thoughts, decisions, etc.? So, in closing, while these advancements within the realm of technology and biology are exciting and have the potential to do a lot of good, it's also important to consider the potential ethical, legal, social, and moral ramifications of such unprecedented science. So, in closing, we were able to confirm that brain organoids can run on computer chips, meaning that computers can run on brains, and Neuralink also proves that brains can run on computers through a direct trial. Thank you!